So today I'm going to talk about the horizontal gene transfer, conjugation, transformation, and transduction. Uh, in my last lecture, we discussed about the pathogenicity island, genomic islands, which is the part which comprises the pathogenic island, ecological island, resistance island. But the question was that how these pathogenicity and genomic islands are being transferred from one bacteria to another bacteria. We also discussed about the mobile elements, where we talk about the TN and IS elements, where the inverted repeats can helps to transfer these segments of gene, a segment of DNA which carries the pathogenicity island from one bacteria to another bacteria, or from one pathogen to another pathogen. Now, what are these methods? What are basically the mechanism of the transfer of these genetic islands, or pathogenicity island, right? So this we'll discuss today. With little more elaboration of some transposons and some mobile elements, including integrons and IS elements. So there are three major processes or three major mechanisms. We call conjugation, transformation, and transduction. So today we'll talk with this process of horizontal gene transfer. Why we call it as a horizontal gene transfer? We call it horizontal gene transfer because there is a transfer of genetic material from one bacteria to another bacteria through extra chromosomal material. So it is not always necessary that the cell divides and transmit the genes of the genomes present on the genome from one bacteria to another bacteria after the cell division. No, it is even without cell division, DNA can be transferred from one bacteria to another bacteria. This we call horizontal gene transfer. It could be plasmid mediated or it could be chromosomal mediated. When it is a plasmid mediated, then it is basically the plasmid transfer, different replicon type plasmid transfer from one bacteria to another bacteria through the process of conjugation, transformation, and transduction. Not transduction, but transformation. And if the genomic part of the DNA is being transferred, it could be through TN elements, through recombination process during the cell division, or through transduction process. So we'll discuss in detail all these three mechanisms which helps the pathogenicity island to transfer from one bacteria to another bacteria. So horizontal gene transfer in prokaryotes is, while in eukaryotes primarily rely on the sexual reproduction for genetic diversity, and prokaryotes have evolved a specialized mechanism for exchanging these genetic material through horizontal gene transfer. So let's talk about the vertical versus horizontal gene transfer. Vertical gene transfer means the transfer of the genome from one bacteria to another bacteria of its own kind, of its own species, because there's a budding, there's a asexual reproduction, right? So the genome of one bacteria get replicated and make a two copy, get divided into two different cells. So we call vertical transfer, whereas horizontal gene transfer is, or we call HGT. It occurs between two independent mature organisms, irrespective of their species and genera. Sometimes, where one is a donor, another is a recipient, without cell division even. Ecological significance of horizontal gene transfer is obvious. That horizontal gene transfer it promote or it facilitate basically exchange of genetic material in prokaryotic, which share the ecological niches, so that it can transfer certain genes which can adapt in certain niches. Example, human gut microbiome, oral microbiome, vaginal microbiome, or they can also, if it out from the it get escaped from certain niches, certain habitat rather, we call them, as because of certain conditions where they cannot survive or they cannot live. So because of some susceptibility or resistance, switching over the genes through this process of horizontal gene transfer. Pathogenicity island, as we discussed in detail in my previous lectures, groups of virulence and coding genes present on the genome or on the plasmid sometimes transferred between the bacteria and this transforming non-pathogenic bacteria into the disease-causing bacteria. So sometimes pathogenicity island through this mechanism of horizontal gene transfer causing the non-pathogenic bacteria to be pathogenic bacteria, right? And antibiotic resistance is another phenomenon, which is one of the, the major cause of antibiotic resistance phenomenon is HGT, horizontal gene transfer, nothing more than that. And it is more of like risky, or it's more of like threatening, if it is through HGT, because in this case, the large number of population in no time can transferred or be transferred through this genetic material or antibiotic resistant genes through plasmid or through DNA elements. The mechanism of horizontal gene transfer is through conjugation and through transformation and transduction, where the conjugation is a process where it's a direct transfer of the DNA, where the naked DNA transferred from one bacteria to another bacteria through bridge-like connection, through pili, and through some other mechanism where they can have tube-like structures which can pass through from after mating these two cells from one cell to another, like this. So suppose these are the two bacteria, right? And say, just take an example of like this. So suppose this is DNA, plasmid DNA. So suppose if this plasmid DNA is to be transferred from this bacteria, we call donor and we call recipient, then this fimbriae may form like tube-like structure, and they can transfer from here to here. It could be through replication process, so that you can have remain one genome part in one bacteria in the donor and another one is recipient. But sometimes if it is non-replicative, then it could be lost from here, and only it is transferred from one bacteria donor to recipient. So this mechanism is called the conjugation process. Now, whereas the transformation process is something different, where the uptake of the naked DNA fragments from environment. So suppose if this is the bacteria, right, and the DNA is from outside, you can have in test tube or some cuvettes or some microfiche tube. Then somehow through experiment, doing some transformation experiment, we called transformation experiment under the laboratory condition, we transfer this DNA here like this. So this is called when the naked DNA is transferred from environment. It could be any environment, then we call the transformation. Whereas the transduction is a process where the bacteria, so this is the bacteria, this is the bacteria virus phage, and this DNA, this is the genomic DNA, is transferred here, and the bacteria may carry the another DNA of the phage. So this phage DNA, after getting into the bacteria, either undergo the lysogeny or undergo lytic induction. So if it undergo the lysogeny, then it get integrated into the bacterial genome like here. Now, when the lytic induction takes place, this DNA get integrated and also carries some part of the bacterial DNA and get incorporated into the phage DNA again, phage particles here. So the bacterial DNA, P plus B, phage DNA and bacterial DNA, both are combined, and it can again attack on another bacteria to transfer in this genome. So this process is called the transduction process. Now these three mechanisms are involved in the transfer of these gene and transfer of these genetic islands in the process of the horizontal gene transfer. what is the fate of transferred DNA? This is very important. Now, when an when an exogenote enters, like from genome from outside enter into the bacteria, what is the fate? So, temporarily it can have like some like may present in outside the genome, or it may get integrated into the genome, right? So, if it is integrated, we call exogenome pairs with the homologous chromosomes, it can undergo the recombination process also, right? For example, if you have, for example, here, this is exogenome, here the endogenome, this is the internal part of the bacteria. Now, the conjugation process takes place to merozygotes, and then integration of the exogenome into the bacterial genome. Here, the red one is a outside genome, and blue one is the inside genome of the bacteria. They get integrated. Now, in case of exogenotes self-replicated, for example, if it undergo, like it could be a phage DNA, it could be the plasmid DNA, it has a capacity to replicate its own. And so if it is replicating using the host machinery or its own independent gene, then they can have another independent copy, like the red one, this one. Now, this is called recombinant cells, and this is reproduction takes place, and the population stable recombinants are formed here after the cells form a number of copies of these plasmids and the genomic DNA. Now, exogenote cannot self-replicate sometimes, so in that case, it is not a stable recombinant process. It could be degraded, like host restriction enzymes can degrade the whole exogenote. So, they are both possibility, either it get integrated and get replicated and protected. So, or it could be, it could not be integrated and it may be lost or degraded. But autonomous replication is quite possible in many cases when the origin of replication is present in the exogenote. The exogenome, like a plasmid genome or the genome which comes from outside, could be a phage DNA which is supported by the phage processing or phage prion particles formations. Phage particle formation into the genome that can carry the DNA of the bacteria after recombination process, after binding with that through temperate phage or through lysogenic stage, and they can carry the phages or acquired the bacterial DNA to infect another kind of cell. So these processes are quite possible in both the cases. Now, the exogenote remains in the cytoplasm but cannot replicate. It's eventually lost during the cell division. See here, it degraded. If it is not, it get integrated. Now, the destruction, that is the host restriction enzymes which are present, which always defend the host from outside DNA, and that DNA is degraded through the host restriction enzymes, are nucleases which are present in that. So the fate of this transferred DNA is either integrated or it get degraded. If it is integrated, it can be replicated, or it can also be replicated if it has origin of replication. So they can have a number of copies are formed and they can be transferred from progeny to next progeny. Now, recombination at the molecular level. What is the molecular basis of this recombination? This is what we have to discuss today. Although the prokaryotes and eukaryotes implies different processes to create recombinant organism, the molecular mechanism of recombination are remarkably similar across the domain of life. Now, the three types of recombination process takes place. One is homologous recombination process. Now, why we talk about the recombination process? Because recombination process means like a breaking and reunion of the DNA. Now, why a breaking and reunion of the DNA is required here in horizontal gene transfer? Because in horizontal gene transfer, you have the genome, as I have just talked about, discussed that, and this genome is get broken, and another genome of outside genome, exogenote, could be phage, could be plasmid, or could be of any DNA, get integrated here, and then the whole genome, hybrid genome or recombinant genome are formed either through transformation or through its own development, own expression of some new proteins for sustainability. For example, various resistant genes are being incorporated. So the bacteria has become resistant against many antibiotics, and they can be protected from the effect of the many antibiotics in the environment, maybe in infection site, or maybe in the sewage water, maybe in some other site. So this is the advantage, and this is through recombination process. All horizontal gene transfer, whether it is a transduction, transformation, or recombination, they all through recombination process if they get integrated. And if it is not integrating, then it is a complementation process. So there's a two process of final effect of the horizontal gene transfer is either complementation or recombination. Now, horizontal gene transfer through homologous recombination, that is exchange of DNA molecule with the same nucleotide sequences, that is called homologous recombination process. It requires extensive sequence similarity, like 100% or 99% similarity required to be. So more is the similar the DNA, higher is the frequency of the recombination process. This is the simple rule, and can occurs anywhere on the chromosomes. It can occurs in any chromosome, sister chromosomes, two chromosomes together if they have similar sequences, or extra chromosomal material, two chromosome, all possible. Now the site-specific recombination is slightly different from the homologous recombination. In site-specific recombination, you have a specific site to bind, to break, and to reunion. I mean, sometimes there's a confusion between homologous recombination and site-specific recombination, but I call all site-specific recombination, even site-specific recombination, all site-specific recombination are even, I call them as homologous recombination. Or in other words, homologous recombination could also be site-specific recombination, because anyway, there is some specific site where it does bind, right? So this is kind of a relative words, and this is kind of a, but more specifically, site-specific recombination is more referred to a sites like in phage. You have at P and at B sites. So other than at P and B sites, no other sites get integrated or get involved in the integration of the phage DNA or phage genome into the bacterial genome. So this is how we call it site-specific recombination process. So it occurs at a specific sequences recognized by specialized enzymes or proteins to guide that integrations or recombination. Generally, it is important in the viral genome, which integrate into the host chromosome, retroviruses, phage DNA, and others. Now, what is transposition? Transpositions is basically the movement of DNA segment that is transposed from different location. It could be within the chromosomes or along, for example, from one part of the chromosomes and another part of the chromosome sometime, or it is from one chromosomes to other chromosomes, or from one bacterial genome to other bacterial genome. So all possibilities are there depending upon the situation and the requirement of the host or of the bacteria. It does not require any homologous sequences because it has some specific site sequences where it does bind. For example, inverted repeat sequences, or it has some other sequences where it has some interaction and some link and some have compatibility of binding of the genome to go for interaction, to go for integration, or in episomal state or temperate state. Now these mechanisms create a new combination of genes. Why we call this a new combination? Because the genes can have combined with some other genes, could have different expression or different proteins, right? So this is how they evolve. New mutations are formed in prokaryotes and species and enables the adaptations to diverse environment. Now let's have a look at this picture. This is the double-stranded break models. I'm not going to talk about detail about the homologous recombination, but just to have a link, because without understanding the homologous DNA recombination or homologous recombination or site-specific recombination, it is very, very difficult to understand the horizontal gene transfer, because ultimately the horizontal gene transfer fate is either it go to recombine or it remain outside. If it is remain outside, it could be plasmid, can replicate, self-replicating. It could have cured at some time, but if it is not like that, then it could have sometime degraded, as I have shown earlier. Now look at this double-strand break. So homologous recombination, we have this is double-strand break here in this strand and this strand, and then it undergo the strand degradations via REC-BCD. So the enzymes REC-BCD is involved in recombination process, and these enzymes are basically involved in nicking, or it has endonuclease activity and the nick at two sites. Then the other side form a D loop and one of the sites is get penetrated inside this D loop like this, right? And when the D loop is formed, it separate the hydrogen bonding between the complementary other strand, that is the fourth strand, and then it form the binding like this, and then it gets separated from here by forming chi site or by forming some other. So there's a unwindings also takes place, like one of the enzymes of REC BCD complex is D, which has a helicase activity and which unwind the process of this. And when unwinding takes place, these two strands get, after cutting, these two strands get separated. So it could have formed like this non-recombinant molecules. It could form recombinant molecule where all the strands get integrated in this case. So this is called recombinant molecule. This is called non-recombinant molecule, because in this case all strands are not involved in the recombination process, whereas in this case all strands are. So it depends on what is the plane of a cut to recombine, right? So if it is on this plane, it form the crossover site, if it form the recombinant molecule or recombinant chromosomes. If it cuts like in this way, it form the non-recombinant molecule, because we call them recombinant because all the 4 strands of the 2 DNA molecules get exchanged their genetic material, that exchange their stretch of DNA. So we call the recombinant molecule. So the key characteristics of these homologous recombination is it occurs anywhere on the chromosomes. It's not required like in a specific region of a chromosome. It results for DNA strand break and reunion, as I have shown earlier here, like this. And then it requires REC genes like REC B, C, D genes, especially REC A proteins involved. REC A protein is basically involved with bind to the single-stranded DNA which cut, and it carries single-stranded DNA by binding in the cavity to take it to the other site where it required to be bind as a double-stranded DNA in other DNA strand. Now, this is ATP dependent reaction. The function is both genetic exchange of a DNA repair mechanism is involved. A post-replication DNA repair mechanism is also involved. The REC BCD system, especially REC A system, to carry the stretch of DNA required to bind to site of lesion. And double-strand break is most widely accepted explanation for how homologous recombination occurs in the molecular level in the bacteria. Now, if you look at this table, you find like BCD recognizes the double-stranded break and generate the single-strand lesions at the break site, where the single-strand binding proteins prevent the like BCD degradations of these enzymes of this DNA molecule. Then REC A is a promote the invasion and displacement because it carries the single-stranded DNA molecules to the D loop, like here, from here. And then RAV-ABC is another protein complex where it involved in the resolvase activity, and where the junction is formed like this. This is called Holiday junction. Holiday was the scientist who identified this junction. It's like chi-like structure, like this, like this, like this. So this is the junction where it may cut like this, it may cut like this. So it depends on what is a cut to cleave this through RAV-ABC complex. It form either recombinant molecule or it form either non-recombinant molecule. Now, non-homologous recombination is basically non-homologous recombination is basically involved in the site-specific recombination, where the 2 sites are independently recognized each other through certain involvement of certain proteins, which facilitate, we call the structural protein on both the host and viruses. If it is our host are both the organism through which the exchange is taking place, or both the DNA where the exchanges are being taking place. So these specific sites are basically required to involve in. So the whole chromosome, any site of the chromosomes may not or should not undergo the recombination process through site-specific recombination, but the very specific site, conserved regions or specific regions are required in binding. So this type of recombination occurs between a specific DNA sequences. Integration of a viral genome into the host chromosome is one of the examples for this site-specific recombination process. Resolution of a circular DNA dimer during the replication process. So this is also specific site-specific recombination process. Inversion of a DNA segments to regulate the gene expression. Suppose if you have a as I talked about the inverted repeats and direct repeats, in the inverted repeats are such repeats which are like in opposite orientations. So when the genes are flanked by the inverted repeats, it might cause the inversion of a gene like this. If this is the gene, and if it is flanked by the inverted repeats, then gene is inverted like this. So this we call inversion process. Now this inversion process help in gene regulation. Now how it helps in gene regulation? It helps in gene regulation because suppose if this is a promoter like this and gene expression takes place like this. Now this is the codon site where the initiation codon of the ORF, where the initiation takes place and the protein expression takes place after mRNA synthesize. Now if the inversion takes place like this, then the promoter goes like this way, in this way, in this way, and the initiation site goes in this way. Now when the initiation site on the this side, promoter remain, because promoter is not going to change, then the expression will not take place because promoter is here at the termination site. So no enzyme will bind here to process the transcription process, and this is called the gene suppression or silencing of the gene, and that's how we regulate the gene through. And it's very interesting that we can have this kind of a regulation system even without having the involvement of any internal factors. Even we remotely do that by some mechanism we do through gene manipulation. So if you look at this, so this is the donor side, the association of the homologous segment, this is red one, and AB, we call this is small ab, we call the host site, where the integration of the donor DNA is being integrated into the host DNA. So it may be phage DNA, it may be bacterial DNA. Now the strand separation takes place in either of the pair of the strand here like this red one, and endonuclease NICS takes place. Endonucleases are the enzyme which cleave one of the sites. It could have break at the single stranded, it could have break on both the strands. It depends on the double strand break or single strand break. So recombination is defined on the basis of the double strand break and single strand break in both the strands, opposite strands which are undergo recombination process. Now the endonuclease NIC host strand takes place, and then the gap is filled by the ligated through this. After the cutting from this, it will bind here. Now once it bind, it can separate it after the replication process, and you can have the heteroduplex formation takes place. Now this is called the recombinant molecule, where one of the strands get recombined, right, at very specific sites. Now in the next replication cycle, this strand, that is, say for example, this is red one, this is the blue one, from this is another strand, and this is, so this form like this form like this. So there's a difference between these two molecules. So this is how the evolution or the mutations takes place over a period of time. So transposition is another important parameter where which is also involved in the process of recombination, or it could be site-specific recombination, because transposon is a site-specific recombination because each TN elements or IS element has specific sites, and these specific sites are not present all across the chromosome or all across the genome, but they are very specific in certain site. Now having that, it can bind to that specific site before they get integrated through the sequences, like either inverted sequences by inversion or by the deletion or insertion through direct repeats. So the key features which includes the transposition process are the genetic element mobility, or we call the genetic mobile element process. It occurs at many sites in the genome. So it mediated by the transposase enzyme, although it has a multiple sites, but the sites are there, the specific sites are present. It often leaves the target sites duplicated. So it could be duplicated, so it can undergo replication process to form another site, or it can transfer from one site to another site also. It creates mutations and rearrangements. So in one it called deleted, in another one it might insert it. So both the way it can work. this is the transposable elements. Transposable elements are we call jumping genes, and these transposable elements are DNA segments that can move and integrates into different sites of chromosomes, from one chromosomes to another chromosomes, and within that. And that was first identified by Barbara McClintock, who discovered the who discovered in 1940, and he got a Nobel Prize in 1983, almost 40 years, 4 decades after the discovery in the maize genetics. So the maize seed was found to be mosaic structure. So you know the maize is, this is a maize, for example, bhutta, right? So this maize were found like a mosaic structures, different type of, I don't have a different color, but I think now she found that why this mosaic structure is formed from the single colored from multiple colored mosaic structure, and that led her to create some new hypothesis and to understand the mobility of these genes from one genome to another genome. During the recombination process, it can recombine so many genetic elements, and she discovered these elements. We call them as a insertion sequences or genetic elements. So insertions, these are inserting within the gene to cause mutations, activating the or inactivating the nearby genes by insertion, disruption, inversion through with reference to the promoters, as I have just told you, and causing the chromosomal rearrangement process. And this transferring the genetic material between the DNA molecules, and these mobile genetic elements have been intensively studied in bacteria and play a very, very important role in bacterial evolution and adaptation, especially in drug resistance and genetic and what you call the antibiotic resistance play a very, very important role in spreading this resistance process in mechanism in this bacteria. With this, thank you very much, and now we have another lectures on soon on continuation to the same elements on insertion sequences.